Hello student, all of you are most welcome on my channel, that is Liberty Solution. In my previous video, I have discussed about chapter number 13, photosynthesis in higher plant. In today's video, I will be discussing about chapter number 14, that is respiration in plant. For better understanding, first you complete photosynthesis and then watch this video. Like, comment, share it with your friends, and don't forget to subscribe my channel to get many more videos. These videos are useful for your school examination, board examination, as well as NEET examination. So let us start to learn respiration in plant. So we'll be doing what? Respiration in plant. Whether it is plant or animal, whenever they respire, that it is plant or animal, when there is a respiration, there is what? Use of oxygen. Because there in respiration, there is oxidation of some material. So in respiration, plant also use oxygen. During photosynthesis, plant use carbon dioxide and they release oxygen. But during respiration, whether it is plant or animal, they use oxygen. Now, what is respiration? It is oxidation of food material. Food material means breaking of carbon to carbon bond of complex molecule. See, this is glucose. It is a complex molecule. There are 6 carbon. There are 6 carbon. These 6 carbons are joined together and there is a bond between these carbons. So when there is this bond is broken, because of oxidation, that is called respiration. It takes place within the cell. This is a cell, within cell. There is some cell organelles are there, like mitochondria is there, cytoplasm is there. So some part of oxidation takes place in the mitochondria and some part takes place in the cytoplasm. And then when there is oxidation of food material, there is a release of energy for ATP synthesis. It is called cellular respiration. This energy is used for absorption. The energy which is released in the form of ATP, this energy is used for various functions like absorption. Suppose there is a plant is there, the plant they are having root, so they will be absorbing water. So to absorb water, there will be use of what? ATP. They use it not only for absorption, but also for transport, movement of material, reproduction, and breathing, and many more. In simple way, we can write the equation of respiration in this way, like glucose undergo oxidation, and the glucose is getting oxidized, and it is converted into carbon dioxide, water is released, plus energy is produced in the form of ATP. Ultimate source of food that is respired is photosynthesis. So whatever food is under respired, that is the product of photosynthesis. So there is a photosynthesis already you have learned. There is some chloroplast. Chloroplast are there. Within this, there is what? There is a photosynthesis takes place. During this, glucose is synthesized and oxygen is being released. This glucose and oxygen again combined together. There is oxidation of this glucose, and then it is converted into what? Carbon dioxide, water, and energy is released in the form of ATP. This is called cellular respiration. Compounds that are oxidized during respiration are called a respiratory substrate. So here glucose is a respiratory substrate. Like some example of respiratory substance are carbohydrate. It is most common respiratory substrate. Proteins, fats, and organic acids, they are also respiratory substrate. The energy released is not used directly, but it is used to synthesize ATP. When there is a oxidation, energy is released. It is released in the form of ATP. When energy is needed, ATP is broken down, hence ATP acts as energy currency of the cell. So ATP is what? Work as what? Energy currency of the cell. Now breathing in plant. Breathing means exchange of gases. So for respiration, plant get oxygen and give out carbon dioxide. In respiration, oxygen comes inside plant body and carbon dioxide is released. In plant, gas exchange occurs via stomata and lenticels. On these leaves, there are tiny pores. These are called stomata. These are the source of what? Gaseous exchange. So carbon dioxide comes and oxygen is released during photosynthesis. And during respiration, this oxygen will be coming inside and carbon dioxide will be released. So these stomata, sometimes they are opened and sometimes they are closed. So on these leaves, there are stomata there, but on other parts, there are what? Some tiny pores are present. They are called lenticels. So on this stem, you will find lenticels. On leaves, you will find what? Stomata. Plant, like in animal, we are having what? This respiratory system, we are having nasal passages, then we are having trachea, bronchi, bronchioles, air sac, lungs. All such systems are not there in plant. Why? Because each plant part takes care of its own gas exchanges. So gas transport is very limited. Gas exchange is very low as compared to animal. Leaves are adapted for maximum gas exchange during photosynthesis. During this, oxygen is released within cell. Most living cells have contact with air. So most of the living cells which are there in contact with the environment. Stem living cells are organized in thin layer beneath the bark. They also have lenticels. And after this bark, there is what? Dead cells are there. So they don't need oxygen. In leaves, stem and root, parenchyma cells are loosely packed that provide interconnected air spaces are there. Therefore, they don't need any special type of respiratory organ. So, complete combustion of glucose yield energy, most of which is given out as heat. This means glucose react undergo what? Oxidation and produces carbon dioxide, water, and energy. This energy is utilized to synthesize other molecules. This energy, ATP, is used to synthesize other molecules required by plant. During the glucose catabolism that break down of any big molecule into a smaller molecule. So during the glucose catabolism, not all the liberated energy goes out as heat. Glucose is oxidized in several small steps. It is here, glucose is oxidized into, there are many steps are there, 1, 2, 3, so many steps are there. It enables some step to couple release energy to ATP synthesis. During respiration, oxygen is utilized and carbon dioxide, water, and energy are released. Certain organisms are adapted to anaerobic condition. They don't need oxygen. Some are facultative anaerobes and others are obligate anaerobes. Now we'll be discussing glycolysis. It is also known as EMP pathway. So what is glycolysis? It is the partial oxidation. Oxidation means breakdown of glucose to two molecules of pyruvic acid in the absence of oxygen. So what is glycolysis? There is a glucose. Under many steps, glucose is converted into two molecules of pyruvate or pyruvic acid. That is same. So this is called what? Glycolysis. So from one molecule of glucose, from one molecule of glucose, two molecules of pyruvic acid or pyruvates are produced during glycolysis. Now the question is, it takes place in where? So it takes place inside the cell, and inside cell there is a cytoplasm. Cytosol means cytoplasm. So glycolysis takes place in the cytoplasm of the cell. For glycolysis, oxygen is not required. It occurs in cytoplasm of all living organisms. Its scheme was given by Embedden, Meyerhoff, and Pernal. So it is also known as EMP pathway. What is the other name of glycolysis? EMP pathway. Here E stands for Embedden, M stands for Meyerhoff, and P stands for Pernal. In anaerobes, it is only processed in the respiration. In anaerobic animals or plants, there is glycolysis is only step of the what? Respiration. There is no, this reaction is not part of anaerobic. Now, in plant, glucose is derived from sucrose. Plant, that is the end product of photosynthesis, or from storage of carbohydrate. So, glucose is obtained from the sucrose. Sucrose molecule, in the presence of enzyme invertase, converted into glucose and fructose. Sucrose is converted into glucose and fructose by an enzyme called invertase. These two monosaccharide, this is also monosaccharide, glucose is also monosaccharide, and this fructose is also monosaccharide. Readily enter glycolytic pathway. Now, glucose and fructose in the presence of enzyme hexokinase converted into glucose 6-phosphate. So it is then isomerized to produce fructose 6-phosphate. And this glucose 6-phosphate undergo isomerization and is converted into fructose 6-phosphate. So this first step, glucose in the presence of enzyme hexokinase converted into glucose 6-phosphate, and glucose 6-phosphate undergo isomerization and converted into fructose 6-phosphate. Glycolysis, EMP pathway, there are what? Many steps of glycolysis. It includes 10 steps under the control of different enzymes. ATP is utilized as 2 steps. In the conversion of glucose into glucose 6-phosphate, here glucose is converted into glucose 6-phosphate. So at here ATP is being utilized and ATP is converted into ADP. Then at step number 3, in fructose 6-phosphate is converted into fructose 1,6-biphosphate. Again, here there is a utilization of ATP. So in first step, what happened? Glucose, which carries 6 carbons, converted into glucose 6-phosphate. Glucose undergo phosphorylation and glucose is converted into glucose 6-phosphate. Then glucose in step 2, glucose 6-phosphate undergo isomerization and converted into fructose 6-phosphate. This fructose 6-phosphate again undergo phosphorylation, and it is converted into fructose 1,6-phosphate. 1,6-phosphate. Step number 4, fructose 1,6-biphosphate, that is contains 6 carbon, splits into PGAL and DHAP. PGAL stand for phosphoglyceraldehyde. PGAL means what? Phosphoglyceraldehyde. And DHAP stand for dihydroxyacetone phosphate. This PGAL contains 3 carbon and DHAP also contains 3 carbon. They are interchangeable. Now this takes place in 5th step. PGAL, interconversion of PGAL into DHAP and DHAP into PGAL. That is step number 5. Step number 6, PGAL, which stands for that is what? Phosphoglyceraldehyde. Phosphoglyceraldehyde is converted into BPGA. BPGA stands for 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. Here, one molecule of NAD is reduced into NADH. And there is a release of hydrogen and a proton. Then step number 7, BPGA converted into PGA. PGA means 3-phosphoglycerate acid. It also contains 3 carbon. Here there is what? Dephosphorylation, and means ADP is converted into ATP. From one molecule, 1 ATP will be produced. So there are 2 molecules, so 2 ATP will be produced. So the ATP which are utilized in step number 1 and 3, 2 ATPs were utilized, and here they are what? Produced. So it is what? Balance. Step number 8, PGA is converted into phosphoglycerate. ATP is also formed when phosphophenol pyruvate PEP convert to pyruvic acid. Then phosphoglycerate converted into PEP. PEP stand for phosphophenol pyruvate, and ultimately phosphophenol pyruvate. When one more important is here in step number 9, there is a what? Release of water. There is release of water. When phosphoglycerate is converted into PEP, PEP stands for phosphophenol pyruvate, water is released. Then PEP undergo dephosphorylation, means ADP is converted into ATP, and ultimately 2 molecules of pyruvic acid are produced in step number 10. So pyruvic acid, so in glycolysis, 4 ATP molecules are directly synthesized from 1 glucose molecule. 2 here and 2 previously. So 2 plus 2, 4. 4 ATP molecules are directly synthesized from 1 glucose molecule in the glycolysis. And how many are used? 2 are used and 4 are produced. So net balance is 2 ATP are released during the process of glycolysis. This pyruvic acid is the key product of glycolysis. Its metabolic fate depends on cellular need. Now, what will happen to this pyruvic acid? If inside the cell oxygen is used, then it will be converted into different product. then it will be undergoing what? Inside the mitochondria. If a plant or animal is what? Anaerobic, then it will get fermented. Means this is the total step of glycolysis, glucose 6-phosphate. First step, glucose 6 is converted into glucose 6-phosphate. Then second step, glucose 6-phosphate is converted into fructose phosphate. Then fructose 6-phosphate is converted into fructose 1,6 biphosphate. Now it will be splitting into triose phosphate, that is what? Glyceraldehyde 3-phosphate, PGAL, and then another molecule is they are triose, means they are containing 3 carbon. That's why they are triose, and they are having phosphate. That's why they are known as phosphate. So one is what? PGAL, and another is what? DHAP. Both are what? Triose phosphate, and they are interchangeable. DHAP stands for dihydroxy acetone phosphate. Then one molecule of NAD will get reduced into NADPH, and now then 2 molecules of triose biphosphate will be produced, and this is what? Nothing, BPGA, means 1,3-biphosphoglyceric acid. Again, this molecule, here there will be dephosphorylation, then ATP will be produced. So this 1 phosphate will be removed. So bi will be removed. So it will become 3-phosphoglyceric acid. 1,3-biphosphoglyceric acid will be converted into 3-phosphoglyceric acid. Again, this will be 3-phosphoglyceric acid, that PGA will be converted into 2-phosphoglycerate, and there will be release of water molecule. Then it will be converted into phosphophenol pyruvate, and here again this phosphate will be released and it will be converted into pyruvic acid. So overall step, from the glucose, what is produced? Pyruvic acid is produced. From 1 molecule of glucose, 2 molecules of pyruvic acid is produced in a multi-step process. This is overall step of glycolysis. Now lactics. Now pyruvic acid is produced here. Now if in different cell, pyruvic acid handled in 3 ways: lactic acid fermentation, alcoholic fermentation, aerobic respiration, that is what? Krebs cycle. If oxygen is there available, so then it will be entering into Krebs cycle, that is called aerobic respiration. If there is a shortage of oxygen, then pyruvic acid will be converted into lactic acid. If there is no oxygen, then pyruvic acid will undergo alcoholic fermentation. So these are 3 phases. In different cell, pyruvic acid is handled in 3 different ways. First is what? Lactic acid fermentation. Second, alcoholic fermentation. Third is aerobic respiration. Now we'll be discussing about anaerobic respiration, that is fermentation. It is the incomplete oxidation of glucose under anaerobic conditions. So if oxygen is not there, there will be incomplete oxidation. It occurs in many prokaryotes and unicellular eukaryotes. So there are 2 types of fermentation. One is what? Alcoholic fermentation, and there is what? Lactic acid fermentation. So these are lactic acid bacteria. These are the yeast. These are fungi. Now, what is alcoholic fermentation? Glucose under many steps is converted into ultimately pyruvic acid. So this is pyruvic acid. Then if oxygen is not there, anaerobic condition is there, then pyruvic acid is converted into ethanol and release carbon dioxide. Here there will be use of NAD. NADH will be getting oxidized into NADI. Here the pyruvic acid, so what is alcoholic fermentation? Here the pyruvic acid formed from glucose is converted to carbon dioxide and ethanol. The enzyme pyruvic acid decarboxylase and alcohol dehydrogenase catalyze this reaction. Example, yeast. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. So sugar is there. Sugar is converted into ethanol. Carbon dioxide releases energy by the yeast. So here, net ATP production for fermentation of one glucose molecule is 2. 4 ATP from glycolysis minus 2 ATP utilized. So there is a production of only 2 ATP. Lactic acid fermentation. Now what is lactic acid fermentation? Here, pyruvic acid is converted to lactic acid in some bacteria. In animal, when oxygen is inadequate during exercise, pyruvic acid in muscle cells is reduced to lactic acid by lactate dehydrogenase. So net ATP production from fermentation of one glucose molecule is 2 here. Reducing agent NADH plus proton is reoxidized to NAD in both type of fermentation. Whether it is alcoholic fermentation or lactic acid fermentation. This reducing agent, that is NADH, is reoxidized to NAD. Now drawback of fermentation. Energy production is limited. Less than 7% of energy in glucose is released and not all of it is trapped as high energy form. Hazardous product, acid or alcohols are produced. Now aerobic respiration. It is a complete oxidation of organic substance in the presence of oxygen, releasing carbon dioxide, water and energy. It occurs in mitochondria. For this, the pyruvic acid, a pyruvate, final product of glycolysis, is transported from the cytoplasm into mitochondria. So, glycolysis, 2 ATPs are produced. Then in Krebs cycle, carbon dioxide is produced. And then there is electrons transport system. 36 ATP are produced and water molecules are also produced. And here oxygen is consumed. So in alcoholic fermentation, lactic acid fermentation, only 2 ATPs are produced. But here in aerobic respiration, 36 ATP are produced here plus here. So the total 38 ATP are produced. There is a big difference in the with respect to production of ATP in aerobic respiration as well as anaerobic respiration. Now, crucial event of aerobic respiration. Complete oxidation of pyruvate by, there is a complete oxidation of pyruvate by stepwise removal of all the hydrogen atoms, leaving 3 carbon dioxide molecules. It takes place in the matrix of mitochondria. So this is a matrix of mitochondria. This is the outer membrane. This is inner membrane. This is intermembranal space. There is a DNA is there, granules are there, crystals are there, ribosomes are there. Passing on of the electron removed as part of hydrogen atom to molecular O2 with simultaneous synthesis of ATP. It occurs on the inner membrane of mitochondria. So ATP synthesis takes place in the inner membrane of mitochondria. This is inner membrane of mitochondria. Oxidative decarboxylation. Pyruvic acid enters mitochondrial matrix and undergo oxidative decarboxylation in presence of pyruvic dehydrogenase. So pyruvic acid plus coenzyme A, CNAD, and the presence of magnesium ion and pyruvate dehydrogenase is converted into acetyl coenzyme A, carbon dioxide is released and NADH is released. It requires coenzymes such as NAD and coenzyme A. During this process, two NADH molecules are produced. Here, two NADH molecules will be produced. From two molecules of pyruvic acid. The acetyl coenzyme A then enters tricarboxylic acid cycle, that TCA. TCA cycle, that is stand for, what is TCA? TCA stand for tricarboxylic acid cycle. Tricarboxylic acid cycle, TCA. Or Krebs cycle, other name of this TCA cycle is Krebs cycle, or citric acid cycle was elucidated by Hans Krebs, therefore it is also known as Krebs cycle. What happened here? If you see this pyruvic acid, which contains 3 carbon, converted into acetyl coenzyme before entering in the citric acid cycle or Krebs cycle. Now, first step, condensation of acetyl group with oxaloacetic acid, OA, and water to form citric acid in presence of citrate synthase enzyme. A coenzyme A molecule is released. So this acetyl coenzyme will react with oxaloacetic acid in the presence of this water, and then coenzyme will be released, and this will be converted into citric acid that contains what? 6 carbon. So here it is having 2 carbon and 4 carbon. So it is becoming what? 6 carbon. Citrate is isomerized to isocitrate. So sometimes citrate, then again there are many steps are there. Citrate is isomerized into isocitrate. Then there is a decarboxylation of isocitrate to alpha ketoglutaric acid. Then this citric acid and isocitric acid will undergo decarboxylation. Decarboxylation means there will be release of carbon. So one carbon dioxide. So now this will be having what? 5 carbon, because 1 carbon is released with what? Carbon dioxide. So now it is known as what? Alpha ketoglutaric acid. Decarboxylation of alpha ketoglutaric acid to produce succinyl coenzyme A. So ultimately there will be decarboxylation of alpha ketoglutaric acid. Then it will be converted into succinyl coenzyme or succinic acid. Succinyl coenzyme is converted to succinic acid, and a GTP molecule is synthesized, substrate level phosphorylation. So GDP will be converted into GDP, and succinyl coenzyme will be converted into succinyl coenzyme will be written here, but here it is not showing. So ultimately there will be one product between alpha ketoglutaric acid and succinyl acid, that is succinyl coenzyme. Succinyl coenzyme A. So ultimately, this will be converted into succinic acid. Oxidation of succinate to fumarate and then malate. So ultimately, this succinic acid will be oxidized into succinate, then fumarate, then malate, and then malate will undergo oxidize into what? Oxaloacetic acid, that OAA. At 3 points of TCA, NAD is reduced to NADH. Here, 1 point is here, and 1 point is here. At 1 point, FAD is reduced to FADH. Here, FAD is reduced into FADH. Continued oxidation of acetyl coenzyme via TCA cycle require continuous replenishment of OAA. It also requires regeneration of NAD and FAD from NADH and FADH. So summary equation of Krebs cycle here: pyruvic acid plus 4 NAD plus 2H2O plus ADP inorganic phosphate takes place in mitochondrial matrix, produces 3 carbon dioxide, four NAD, 4 hydrogen ion, FADH and ATP. This glucose is broken down to give 6 carbon dioxide, 8 NADH, 2 FAD and 2 ATP. This is Krebs cycle in detail. Citrate, cis-acotinate, D-isocitrate, then alpha-ketoglutarate, then succinyl coenzyme A, then succinate, then fumarate, then malate, then oxaloacetate. These are the various points. So here there is a release of carbon dioxide. Here also there is release of carbon dioxide. Here there is a release of carbon dioxide. Now we'll be discussing about aerobic respiration, electron transport system, that is ETS, and oxidative phosphorylation. Electron transport system, that is ETS, is a metabolic pathway present in inner mitochondrial membrane through which electron passes from one carrier to another. So these are what? Some carriers are there. So this is one carrier. There is a complex one, NAD dehydrogenase, one carrier, second carrier, third carrier, fourth carrier, and ultimately there is what? Fifth carrier. So these are nothing, they are what? Electron transport, component of electron transport system. So this is what? Inner membrane. This is intermembranal space. So on the inner membrane, these are electron transport component of electron transport systems are there. They are transporting electron. This is to release and utilize energy stored in NADH and FADs. This, what is the use of ETS? ETS is used to release what? Produce energy. ATP, which is stored in NAD and FAD, means there will be conversion of NAD will be converted into ATP and FAD will be converted into ATP in the electron transport system, which are produced during what? TCA cycle. The electrons are passed on to oxygen to form H2O. So electron, these are passed onto oxygen. Then here, there will be release of what? Oxygen. There will be release of what? Water. So this electron will be transported here and then there will be what? Release of water. Electrons from NADH are oxidized by NADH dehydrogenase complex first. So here there are electron NADH, NAD is having energy. So they are having energy means in the form of what? Electron. So these electrons from NADH are oxidized by NADH dehydrogenase. This is what? NADH dehydrogenase. This is a part of complex 1. Then electrons are then transferred to ubiquinone, that is Q located within the inner membrane. Ubiquinone also receive reducing equivalent via FAD. Here there is this ubiquinone, that UQ stand for ubiquinone. This ubiquinone receive electron, then it transport it to it get what? Reduced. Here also it also receive reducing equivalent via FADH. So FAD molecule will enter in transport system over here. That is generated during oxidation of succinic into citric acid. Then reduced ubiquinone, ubiquinol or QH2 is then oxidized with the transfer electron to cytochrome via cytochrome BC1 complex, that is complex 3rd. So there is a complex 3rd is there. Which is made up of cytochrome, complex. Cytochrome C is a small protein attached to the outer surface of inner membrane. It acts as a mobile carrier of electron between complex 3rd and 4th. So this is what? Cytochrome C. It is present between complex 3rd and complex 4th. So it transports electron between complex 3rd and complex 4th. Then complex 4 refers to cytochrome C. Here there is a cytochrome C. Cytochrome C oxidized complex containing cytochrome A and A3. There is a cytochrome A and A3 and 2 copper centers. Here, ultimately when it reaches here, electron is picked by this proton is there. So this hydrogen proton will become converted into hydrogen atom, and this hydrogen atom will react with oxygen and it will be releasing water. When the electron passes from one carrier to another via complex 1st to 4th, they are coupled to ATP synthase complex 5th for ATP production. Number of ATP molecules produced depends on nature of electron donor. So oxidation of 1 NAD gives what? 3 ATP. Oxidation of 1 FAD gives what? 2 ATP. So there are how many complexes are there? 5 complexes are there. Complex number 1, there is NAD dehydrogenase is there. Complex number 2, FADH2 is there. Complex number 3, cytochrome BC1 is there. Complex number 4, cytochrome C oxidase. And complex number 5, it is having ATP synthase. So you have to remember this. From one molecule of NAD, how many ATP are produced? So answer will be 3 ATP. And from one molecule of FAD, how many molecules of ATP will be produced? 2 ATP. Overall electron transport chain, NAD enters into complex 1, then enters into complex 2nd. In complex 2nd, FAD also enter in this electron transport system. Then here complex third, that is made up of cytochrome B, C1. Then complex third and fourth are connected with cytochrome C. Then this from complex number 4, this electron will be will be used by hydrogen ion, will be converted into hydrogen atom, and it will be used to produce what? Water molecule. In aerobic respiration, the role of oxygen is limited to terminal stage. Yet oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as final hydrogen acceptor. In respiration, energy of oxidation reduction is utilized for the phosphorylation. So this process is called oxidative phosphorylation. So electron transport system is also known as what? Oxidative phosphorylation, because oxygen is required here. Oxidative phosphorylation is different from photophosphorylation. Here, light energy is utilized for the production of proton gradient for phosphorylation. So the energy released during ETS is utilized to synthesize ATP by ATP synthase, that is a part of complex 5. ATP synthase has two major components. F1 head, this is what? F1, and then F0. F1, that is head piece, peripheral membrane protein complex, and F0, it is what? Integral membrane protein complex. So F0 is integrated with the membrane, and F1 is what? It is what? Peripheral membrane protein complex. So this will be, there will be passage of hydrogen and proton from outer side to inside, and during that the ADP will be converted into ATP. F1 headpiece, that is peripheral membrane protein complex, site for ATP synthesis from ATP and inorganic phosphate. PI stand for inorganic phosphate. F0, that is integral membrane protein complex. It forms a channel through which proton across the inner membrane. The passage of proton is coupled to the catalytic site of F1 component for ATP production. For each ATP produced, 2 hydrogen ion passes through F0 from the intermembrane space to matrix down the electrochemical proton gradient. So sometime question will be there, for the production of 1 ATP, how many hydrogen ion are passes through F0? So 2 hydrogen ions are passes for the production of 1 ATP. So this is all about what? Aerobic respiration. So this is what? Cytosol. This is outer mitochondrial membrane. This is inner mitochondrial membrane. In the intermembrane space, there is a large number of protons are there. These protons are entering in this membrane from the matrix of the mitochondria through these various complexes. The complex 1 is there, 2 is there, 3 is there, 4 is there, 5 is there. So here the electrons are transported by these complexes. Complex 1, ultimately they reaches to the what? Complex 4, and then ultimately there is what? These hydrogen ion are picked up by here. They neutralize this proton, and ultimately water is being produced. And then these hydrogen ion, which are the proton, which are present in intermembrane space, they produce what? Proton gradient. And because of that, this ATP synthase enzyme become activate, and they start passing from intermembrane space to the inside. So ultimately, ATP will be produced. For production of 1 ATP molecule, 2 proton need to be passed through from intermembrane space towards what? towards matrix. So in glycolysis, glucose is converted into pyruvate. So two ATP molecules are produced. Then there is what? Then pyruvate enter into acetyl coenzyme. Neo ATP is produced. So this is what? This is link reaction. Then acetyl coenzyme enters into Krebs cycle. So how many ATP are produced? 2 ATP are produced here. And then through electron transport system, 32 ATP are produced. 32 ATP are produced. So net gain of ATP from each glucose molecule is calculated based on the following assumption. All steps in glycolysis, TCA cycle and ETS occur sequentially and orderly. The NADH synthesized in glycolysis, it transferred into mitochondria and undergo oxidative phosphorylations. Intermediates in the pathway are not used to synthesize other compounds. Only glucose is being dispersed. Other alternative substrates are not entered in the pathway at any stage. Such assumptions are not valid because all pathways work simultaneously and do not take place one after another. Substrate enter the pathway and are withdrawn from it as and when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means. Such calculations are used to appreciate the efficiency of living system in extraction and storing energy. So comparison between fermentation and aerobic respiration. Fermentation, there is a partial breakdown of glucose. In aerobic respiration, there is a complete breakdown of glucose to carbon dioxide and water. There is in fermentation net gain of only 2 ATP, but in aerobic respiration, there is a net gain of 36 ATP. NADEH is oxidized to NAD rather slowly, and here NADH in aerobic respiration, NADH is oxidized to NAD and very vigorously. Now we'll be discussing about amphibolic pathway. Glucose is favored substrate for respiration. So all carbohydrates are first converted into glucose for respiration. Other substrates are also respired. Fat breakdown into glycerol. This is a fat breakdown into glycerol. Then this fat glycerol enters this in the form of what? Dehydroxyacetone phosphate. It can enter here or it can directly enter here at the acetyl coenzyme A. Fat breakdown into glycerol and fatty acid. Fatty acid are degraded to acetyl coenzyme and enter the pathway. Glycerol is converted to PGL and enter the pathway also. So there are two entry points for fats. One entry is what? Here they are entering, and another is what? They are entering here. Proteins are degenerated. Proteins, there is only one entry point for protein. Fat is having two entry points. Then proteins enter here in the pyruvic acid. Proteins are degraded by protease into amino acid. Each amino acid after deamination enter the pathway at some stage in the Krebs cycle or pyruvate or acetyl coenzyme A. The respiratory pathway is generally considered as a catabolic pathway, but it involves both anabolism, synthesis, and catabolism breakdown. So it is better to call it amphibolic pathway. So there is what? Catabolism and anabolism both in respiratory pathway. That's why it is called amphibolic pathway. Fatty acid breakdown to acetyl coenzyme A before entering the respiratory pathway, but when it organism need to synthesize fatty acid, a still coenzyme withdraw from the respiratory pathway. Similarly, during breakdown and synthesis of protein, respiratory intermediates are involved. Now, respiratory quotient, that is RQR, respiratory ratio. It is also known as respiratory ratio. What is how you can calculate respiratory quotient? The volume of carbon dioxide evolved divided by volume of oxygen consumed. We can calculate what? RQ. RQ is the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed in respiration. So RQ means volume of carbon dioxide evolved, volume of oxygen consumed. RQ depends upon the type of respiratory substrate. RQ for carbohydrate is 1 because equal amount of carbon dioxide and oxygen are evolved and consumed respectively. So here in this molecule, this is a carbohydrate. How many molecules of oxygen are consumed? 6. How many molecules of oxygen are consumed? Here 6. And how many are carbon dioxide evolved? 6. So 6 carbon dioxide are evolved and 6 oxygen molecules are consumed. So RQ value for carbohydrate is 1.0. So RQ for carbohydrate is 1, RQ for fat is less than 1, and RQ for protein is equal to 0.9. RQ for this year, see this is a fat here, 102 carbon dioxide molecules and 145 oxygen consumed. So RQ for this fatty acid is what? 0.7. RQ for protein is 0.9. You have to remember these facts. In living organism, respiratory substances are often more than 1. Pure protein or fats are never used as respiratory substrate. So thank you this much. In this way, this chapter is complete. Now I am signing up. Thank you very much for watching this video.